Hello and welcome. Welcome to Biosignaling 2.1. Previously, in Biosignaling 1.1 and 1.2, I explained the principles of biosignaling and cell signaling, of course, at the introductory level. I talked about ligands, messengers, receptors, some basic features of signal transduction pathways, and so on. In this lecture and in the next one, I'm going to talk about some molecular switches. So the outline for this particular lecture is that I'm going to first introduce molecular switches. Then I'm going to introduce the concept of G proteins. And then I'm going to talk about different types of G proteins and the receptors which are coupled with G proteins. Although I talked a little bit about the G protein coupled receptors at the end of Biosignaling 1.1, in which I talked about the different types of receptors, but we're going to elaborate on that more. And then we're going to talk about some common signal transduction pathways in which G proteins have a key role. So this is the general outline of Biosignaling 2.1. So molecular switches. You know, there are some intracellular signaling proteins which are like switches. I mean, like a switch, they have an on and off state. Okay, so in this case, in the case of biomolecules and proteins in the cell, when we say that they are in on state, it means they are active. And by active, we mean that they can actually make some interactions with they can interact with other proteins like effector proteins, or they can catalyze something if there are enzymes and so on. But when they're in off state, it means they're not active. They can no longer interact with other proteins, or they can no longer function. So this is the concept of molecular switches. There are various kinds of switching mechanisms. There are various kinds of mechanisms in which, by which a single protein is turned on or off. For example, phosphorylation, which is the chemical reaction of adding a phosphate group to a protein or a biomolecule, may make a protein or any biomolecule active or inactive. Okay, so phosphorylation, which is the mechanism I'm going to talk about later in the next lecture, is one of the common mechanisms for switching a protein or any biomolecule on or off. Not any, I mean some particular biomolecules. So this is the concept of molecular switches. Now let's talk about one of the most common molecular switches, which is GTP-binding proteins. So GTP-binding proteins are proteins to which some GTP or GDP molecules are bound. So GTP-binding proteins, they can be also abbreviated as G proteins. I mean, they're commonly known as G proteins. And they're molecular switches. And let's see, what do we mean by a GTP protein, a GTP-binding protein on a state and off state? You know, when the GDP, guanine diphosphate, which has only two phosphates, when GDP is bound, the G protein is switched off. It's not active. But when GTP, guanine triphosphate with 3-phosphate is bound, it's switched on and it's active. There are two major types of, there's a large family of G proteins, but there are two major types. The first one is a monomeric one. Monomeric means something composed of one single whole unit. Okay, so monomeric G protein is just a protein which is composed of one single unit. Also known as monomeric GTP-binding proteins. We have also hetero-trimeric G proteins. In this case, hetero means different. Trimeric means something which is composed of 3 parts. So when we say hetero-trimeric G protein, we're talking about a G protein which is composed of 3 different subunits. Okay. I'm going to explain the exact structure of these proteins. So these are the two main types of G proteins. But in this lecture, I'm also going to talk about some of the regulatory proteins, some proteins which regulate the activation or inactivation of G proteins, which one of them is a GTPase activating protein, or GAP, which actually increase the rate of GTP hydrolysis. Well, back there, I told you that when GDP is bound, the G protein is inactive. So when GAP, GTPase activated protein, facilitates the hydrolysis of GTP to GDP, okay, it turns GTP into GDP, it means that GAP proteins actually make G proteins inactive. And we have another kind of regulatory protein, which is guanine nucleotide exchange factor. Its role is to promote the release of GDP. So what? So that GTP, guanine triphosphate, can bind to the G protein. And again, as I told you, when GTP is bound, the G protein is activated. So the key role of GEFs, guanine nucleotide exchange factors, is to actually activate G proteins. Okay, so I'm going to talk about their mechanisms, and their function as well. Now let's talk about a little bit about the switching mechanism. A little bit more, and let's see what happens when a protein is turned on, when the G protein is turned on or off. So we have a G protein here. Why is it off? Why is it inactive? Because it is bound to GDP. Okay. Now a signal comes in. The signal activates a set of chemical reactions by which the GTP binding happens. GTP binding happens. GDP gets out, okay, and GTP comes in. And what do we have now? An active G protein. Why? Because GTP triphosphate is bound right now. But when the signal goes out, well, we have GTP hydrolysis. GTP is hydrolyzed to GDP and an inorganic phosphate here, and the activated G protein turns back to its inactive state. Okay. So we have a monomeric G protein, a monomeric GTPase, which is another name for monomeric G protein. So when a signal comes in, it certainly has to activate GEF. Okay. So in this case, we said that GTP binding should happen. And as I told you, there are some regulatory proteins. In this step, GEF, or guanine exchange factor, is going to promote the release of GDP so that GTP can bind and make it active, make the G protein active. And for the hydrolysis of GTP, we need another enzyme, GTPase activating protein, or GAP, which hydrolyzes GTP and turns the G protein inactive. Okay, so this is how it turns this is how a monomeric G protein is activated and deactivated when a signal comes in and when a signal comes out or gets out of the cell, and how GEF and GAP activate and deactivate the G protein. But now let's see why that extra phosphate can make GTP active. Okay, what's so special about that extra phosphate that GTP has that makes the G protein active? Well, in this case, you can see a GTP bound state. GTP is bound, and this is the on state. Okay, this is the active state. Now let's see the structure. Sorry, my pointer is. Okay, here this is the monomeric G protein. Okay, it is one whole unit. It has a guanine or nucleotide binding pocket, okay, in which the GTP or GDP bind. binds. And you can see that it has two switches, switch 1 and switch 2. These two switches are parts of the GTP which are going to interact with other proteins and make them active or activate them. So look at their conformation and look at their positions. You know, this gamma phosphate, which is the last phosphate of guanine's triphosphate, is forming some bonds with an amino acid in switch 2, which is glycine 60, the 16th glycine amino acid in switch 2, and also with threonine 35 of switch 1. Okay? And these two bonds between these two amino acids in these two switches and this gamma phosphate, the last phosphate of GTP, is holding, you know, these two bonds are holding these two switches in this active position, okay? So that they can interact with other proteins and activate them or whatever they do. And now let's see what happens. You know, you see these scissors here. Let's see what happens when we cut this gamma phosphate out and hydrolyze GTP into GDP, diphosphate. Now, we no longer have those holding bonds here. We no longer have those holding bonds here. So now, these two switches undergo a conformational change, and they change their position and they change their conformation so that they no longer they're no longer able to make interactions or interact with other effector proteins, with other proteins. So that's why when GDP is bound, the GTP protein is inactivated. You see that they're no longer in this active position. These two switches are no longer in this active position. They are no longer held by these two bonds. Okay, so that's why that one single phosphate causes a G protein to be active. And here you can see a little bit, this is very schematic. This is closer to the real structure of a protein. This is switch 2 and switch 1. You can see the threonine and glycine 60. And here you can see the gamma phosphate. You can see that these two amino acids, the amino acid residues in switch 1 and switch 2 are really close to it and are making interactions with it. But when we cut it out and when we hydrolyze GTP into GDP, you can see that these two switches undergo a conformational change, and you look at the position of threonine 35 right now. It's no longer near the G protein, okay, GDP, okay, it changes position. Okay, so this is how switching happens, and this is how one single phosphate group can turn a G protein active. Okay, let's review what I've just told you. This is the monomeric G protein. Look that it is covalently attached to a farnesyl group. This farnesyl group is, you see this, it has a hydrocarbon tail, and it is covalently attached to the C-terminal cysteine here, and it helps the G protein to attach to the plasma membrane. And this is the G protein in its inactive state. Why? Well, because it is the GDP is bound. And this is the same G protein, but in the active state. Why? Because we have a GTP, okay, GTP bound here. And look at the position of threonine and how it's very beautiful. Look at it. It's threonine 35 here. Okay, this is here, and here we have GDP. Okay, look at the position. It's far from the GDP. And look at this arrow. This arrow shows the conformational change which happens when GTP binds. When GTP binds, thanks to this gamma phosphate or the third phosphate it has, it attracts and forms a bond with this threonine 35, and now look at the position of threonine 35. Okay, it's now closer to the it's forming a bond with the last phosphate, and also the switch 2 as well. And now it can interact with effector proteins. Okay, that's why we call it. We say that it is in the active state. So what happens between these two? Okay, what happens when we have an inactive protein, G protein, and it is activated? Well, first we have a protein bound to GDP. Okay. Now GEF comes into the scene. What it does is that it promotes the release of GDP. GDP is repelled, or is let's say it's repelled. And now we have GTP coming in, and here we have protein bound to GTP, which is the active state. But here we have GAP. GTP is activating protein, hydrolyzing the GTP into GDP and an inorganic phosphate. So the hydrolysis happens here. The extra phosphate gets out, and again we have protein bound to GDP, or the inactive protein. Sometimes in this step, proteins may bind to guanine dissociation inhibitor, GDI. Well, GDI is another regulatory protein of G proteins, but it's not as common as GAPs or GEFs. But what it does is that it prevents the interaction with GEF. So it prolongs or lengthens the inactive state of G protein, which is sometimes needed in receptor desensitizations, for example. Okay? And this is how activation and deactivation of G proteins happens. I'm not satisfied yet. Of course, I'm going to show you the exact, chemical interactions or molecular dynamics of these enzymes. But one point I want to mention here is that switching off is as important as switching on, because when we don't want a signaling molecule in the cell to be always active, because that may cause a lot of trouble for us. Okay, so we always need a mechanism to switch a protein or a biomolecule off as well. Okay. Sorry. Okay. Let's talk about the activity of guanine exchange factor in H-Ras. H-Ras is human rat sarcoma. It's a subfamily of monomeric G proteins, and it has a specific GEF called SOS. So SOS is it's an abbreviation for son of 7-less. 7-less is a group of genes. I'm not going to explain the terminology here. Just know that SOS is an H-Ras specific guanine exchange factor. It has two domains: the Ras exchange motif domain, or REM, or REM domain, and there's an N-terminal catalytic domain, or CDC25 domain. And this is the H-Ras. You can see switch 1 and switch 2 of the Ras. How does this SOS protein, or GF, promotes the release of GDP? Okay, what it does? It does two things. First of all, it inserts two amino acid residues in this guanine or nucleotide binding pocket. Let's see what they are and what they do. The first one of those amino acid residues is leucine. Leucine is a non-polar hydrophobic amino acid. And when it is inserted in this binding pocket, it disrupts the electrostatic coordination of the magnesium ion in this binding pocket. And the magnesium ion in the binding pocket is necessary to coordinate the GDP and GTP, okay? So, and because it is ion, it is electrostatically coordinated by other ions or amino acid residues in the binding pocket. And this leucine, because it is hydrophobic or nonpolar, it disrupts the electrostatic coordination, okay, of the magnesium. So the magnesium is no longer correctly coordinated in the binding pocket. We have also glutamic acid, E942, I guess. Yeah, 942. E942, which is a glutamic acid, is an acidic amino acid residue. Let's see what it does. It has a negative charge. It actually causes the electrostatic repulsion, okay, electrostatic repulsion of the alpha phosphate, or the very first phosphate of GDP. So, thanks to this negative charge, it repels, it causes the alpha phosphate of GDP to be repelled. So it repels by electrostatic repulsion. It repels the alpha phosphate of this GDP molecule. The second thing that this sauce does is that it inserts the alpha H helix into the binding pocket. When it inserts this alpha H helix into the binding pocket, you can see this is switch 1, and you can see that it pushes this switch 1 downward. Okay, so this is switch 1, or this is switch 1. It inserts this alpha H helix, and it pushes the switch 1 downward, okay, represented by this blue arrow pointing downward. Okay, so it pushes the switch 1 downward. So by doing that, it opens the binding pocket. Now the binding pocket is open. The magnesium ion, which is necessary to coordinate the GTP, is disturbed. Its coordination is disturbed. The GDP, the alpha phosphate of GDP, is also repelled by the glutamic acid. What does happen? What happens now? GDP escapes. That's how GEF promotes or facilitates the expulsion of GDP. And now GTP can bind to the nucleotide binding pocket and turns the H-Ras active, makes the H-Ras active again. So this is how a GEF enzyme promotes or facilitates the release of GDP so that GTP can bind and turn G protein on or active. So it inserts 2 amino acid residues. They disturb the electrostatic coordination of a magnesium ion. One of them is a glutamic acid, which repels the alpha phosphate of the GDP, and also it inserts an alpha H helix into the binding pocket. It pushes the switch 1 inward so that the binding pocket is now open and the GDP can escape. This is the activity of GEF. You know, I'm fascinated by these, molecular interactions and the very exact mechanism of proteins and enzymes. You know, 2 years ago when I was still at high school, my biology teachers, they told us that, well, this enzyme does that, this enzyme does that, that protein works like this, but they never told us the exact mechanism. And I'm interested in molecular dynamics, protein dynamics, and how they make interactions with each other, and how by just, a few amino acids, they catalyze a very complicated reaction. And this is fascinating, I guess. So this is the activity of GEF. Now let's talk about the activity of GAP, a RAS-specific GAP, or GTPase activating protein. Well, I reviewed the GAP a lot, but I have to do that again, I guess. It's better to do that. GAP is GTPase activating protein. It promotes it facilitates the hydrolysis of GTP into GDP. Now let's see how does GAP facilitates the hydrolysis of GTP. Here we can see the RAS-specific Jesus. Sorry, my pointer is now, funny. So there's a Raff specific GAP. You can see the GAP domain here. And you can see the H-Ras here. Again, switch 1, switch 2. And here we have GTP bound. So this is the active H-Ras, and we want to make it inactive. So this part is H-Ras bound to GMP, and this is the beta or the second phosphate, and this is the third phosphate. So we have a GTP here, we have a magnesium ion coordinating that GTP with water molecule, and here we have the a part of the GAP. So how does the GAP facilitate the hydrolysis of this last phosphate, this third phosphate? Well, GAP inserts a positively charged arginine finger. Okay, so this is the arginine finger. It inserts this arginine finger, which has a positive charge at the end. Okay, next to this negatively charged gamma phosphate. Putting this arginine finger next to this or close to this gamma phosphate reduces the amount of energy which is required to bring that gamma phosphate into an intermediate position, in which the gamma phosphate, right now it is this gamma phosphate is in that intermediate position. In this intermediate state, it is shared between the donor, which is GTP, and the water, and the receiver, which is the water. So, look at this positive charge. It somehow, to some extent, neutralizes the negative charge of the gamma phosphate, and it also brings the gamma phosphate, or reduces the amount of energy to bring, which is required for bringing the gamma phosphate in this intermediate position between the donor, the GDP, and this water molecule. Since its charge is partially neutralized, and it is right now close to this water molecule, it can be easily transferred, hydrolyzed, and now we are left with a GDP. A GTP is hydrolyzed. Okay, so this is how it works. I made a small model of that, actually, to show you the exact mechanism. I guess now you can see that. I hope you can see that. First of all, I have to apologize because I didn't have a lot of these molecular kits. So my model is not very complete, but it has the essential parts. So this is that arginine finger inserted by GAP into this, GTP binding pocket. This is the gamma phosphate of GTP, and this is that water molecule. This part of the arginine finger has a positive charge, which attracts and neutralizes the negative charge of this gamma phosphate, and also brings this gamma phosphate into this intermediate position when it is shared within the donor, its donor. It's not shown here, but the rest of this molecule is GDP, and this is the last phosphate of GTP, and it's shared between the donor, which is GTP, and this water molecule, so it can be easily transferred to the water molecule, or hydrolyzed, basically. Sorry, not transferred, but hydrolyzed by this water molecule. So all GAP does is to insert this single arginine finger into this binding pocket. Thanks to its positive charge in this part, it brings this, or reduces the amount of energy required for bringing this gamma phosphate into this intermediate position, in which it can be shared or hydrolyzed by this water. That's it. That's all it does. But again, it's beautiful because by doing a very simple, relatively simple task, it facilitates the hydrolysis of a G protein. Sorry, GTP. Glutamine 61, Q61 of the H-Ras, plays a very important role in coordinating the water molecule and also the gamma phosphate. So this is also important here. We have another arginine finger, arginine 903. This coordinates not the arginine finger, but the loop comprising that catalytic arginine finger. So this one is also important. So just wanted to mention these two important amino acid residues and their roles as well. These two amino acid residues have important roles in, you know, in the hydrolysis or coordinating the molecules and the hydrolysis of GTP. So I talked enough about the monomeric G protein. I guess it was enough for an introduction to monomeric G proteins. Now let's talk a little bit about trimeric G proteins. Well, I told you that heterotrimeric G proteins, you know, hetero means different, trimeric means something that is composed of 3 subunits. So heterotrimeric G proteins are G proteins which are composed of 3 different subunits. And well, these are the 3 different subunits here. These are the 3 different subunits here. You can see the plasma membrane here and the 3 subunits here on the cytosolic side, of course. We have the alpha subunit, and we have beta and gamma. The alpha subunit has the GDP or GDP. It also has a Ras domain related to other Ras or GTPase G proteins. And we have an alpha helical domain or AH domain which clamps the nucleotide in the nucleotide binding pocket. Okay, so you can see that the alpha and gamma, not beta, only alpha and gamma are covalently bound to these lipids, and these lipids help them to attach to the plasma membrane. Okay, and when, you know, right now this one is the GDP is bound, so it is the alpha subunit is inactive. But when the GTP binds, the alpha subunit gets active. It becomes activated or active, and we have 2 general types. I mean, we have different types, but there are 2 general types. We have a stimulating G protein, which obviously stimulates effector proteins and make them active. And we have inhibitory G proteins, which inhibit the effector proteins. And in the stimulating G proteins, when GTP binds and this alpha subunit gets activated, the alpha subunit is, dissociates from the beta and gamma. And the beta and gamma, although they don't have GTP or GDP bound to them, but they, when alpha subunit is activated, they are activated too, and alpha and beta, beta and gamma subunits together form a functioning unit, and they can act as transducers and activate and activate other effector proteins. But in inhibitory G proteins, when the alpha subunit of the inhibited G protein gets activated by GTP, it doesn't dissociate from the beta and gamma subunit, but it reorients its position relative to the beta and gamma. Okay, this is what I wanted to mention. So here we have the alpha subunit with its GTP or GDP binding site. We have the beta subunit and the gamma subunit. So now let's talk about the, let's introduce some major classes of these G proteins. Of course, these are the G alpha classes, the alpha subunit of those G proteins, different G proteins. We have a stimulating, the alpha subunit of a stimulating G protein, which stimulates its associated effector, which is the adenylyl cyclase, the same enzyme responsible for the production of CAMP. And because this is a stimulating G protein, it stimulates and activates this adenylyl cyclase, so it increases the production of CAMP. But we have an inhibitory G protein, or the alpha subunit of inhibitory G protein, which again, its effector is adenylyl cyclase, but since it's inhibitory, it would decrease the level of CAMP. But inhibitory G protein, or the alpha subunit of the inhibitory G protein, also has affects potassium channels and potassium channels, and it changes the membrane potential as well. So it has another effector, which is an ion channel. And we have, for example, G alpha Q, which its effector is phospholipase C beta, which is responsible for the production of inositol triphosphate and diacylglycerol as second messengers. We are going to see some of those signaling mechanisms or signal transduction pathways in which G proteins activate those effectors and ultimately result in the production or inhibition of the production of these second messengers. Okay, so let's see how a G protein, a trimeric G protein, is activated. Well, in the case of, in monomeric G proteins, we had a GEF, okay, guanine nucleotide exchange factor, which was responsible for the activation, for the release of GDP so that GTP can bind. The GEF, or its role for trimeric G protein, is played by the G protein coupled receptor. So at first we have an inactive G protein coupled receptor, and we have an inactive G protein complex. When a signal molecule binds to the G protein coupled receptor, the G protein coupled receptor undergoes a conformational change. And that conformational change makes the G protein coupled receptor activated or active. Once it is activated, it's going to activate the G protein or the trimeric G protein. And the main G protein coupled receptor is actually going to cause a conformational change in the trimeric G protein. And that conformational change is shown here it's a displacement of alpha helical domain, okay, AH domain. And when the AH domain is displaced or is opened due to the conformational change caused by the G protein coupled receptor, it no longer covers the GTP-binding protein, the binding pocket. GDP escapes and GTP gets in, okay? The reason that when GDP gets out, GTP replaces it is due to the tenfold concentration of GTP compared to the GDP. So GTP exists in the cytoplasm in higher concentrations as compared to GDP, and also thanks to its third phosphate, or the gamma phosphate, GTP has a higher binding affinity compared to the GDP. Okay, so that's why when GDP escapes after alpha helical domain opens, GTP comes in, and when GTP is bound, we have an activated alpha subunit, and this is a stimulating G protein, because you can see after alpha subunit is activated, it is detached from the beta and gamma subunits. Okay, so it is detached, and now both the alpha subunit and these two beta and gamma subunits as a whole unit, they are going to activate effector proteins. So this is the mechanism of action, and this is how the G protein, a trimeric G protein, is activated. Now let's talk about G protein coupled receptors. There are various kinds. I guess there are the most abundant receptors in organisms, but there are various types of G protein coupled receptors, but they all have a characteristic feature, which is that they're all a single amino acid sequence, and they have 7 TMs, or transmembrane alpha helices, okay, threading back and forth in the plasma membrane with with 4 exoplasmic loops and 4 cytoplasmic loops or cytosolic loops, and we have the N-terminal outside of a cell and the C-terminal inside. So this is the general structure of a G protein copper receptor. This one is a real example of it. Is the beta-adrogenic receptor, which is a G protein copper receptor. And you can see these alpha helices. You can see the C-terminal here, the N-terminal here, and we have the ligand, which is the adrenaline, the hormone adrenaline, here in its binding site. And if you look at this receptor from the top, so this is a top view, and you can see the this ligand or adrenaline in its binding site. You can see that it forms some binds with some amino acid residues or sign chains of these amino acid residues and these loops and alpha helices, and is tightly bound to its binding site. There's a relatively deep binding site in most of the G protein copper receptors. This is also important. It's an important feature of most of the G protein copper receptors. So now let's see. I told you when a signal comes in, or when a ligand binds to a G protein copper receptor, it makes it activated. It makes it active. So you can see here that this is the same beta-2 adrogenic receptor that we saw in the previous slide. This is in its inactive state when there's no adrenaline, there's no ligand bound to it. And this is the G protein, the trimeric G protein, which is coupled with this receptor. And since this is the receptor is inactivated, it's not activated, the heterotrimeric G protein is not activated as well. So, but now we have a ligand. This is an active receptor. We have a ligand here bound to it. Okay, so it does. You can see a conformation change happened, and this conformation change also activated the heterotrimeric G protein, particularly the alpha subunit, and now you can see that the GTP, okay, is going to bind to the alpha subunit and activates that. Okay, so this shows the difference between the active and inactive G protein copper receptor. But in this picture, you see the exact conformational change and the exact mechanism by which G protein copper receptor acts as a guanine exchange factor. Well, when a ligand binds to a G protein copper receptor, particularly this one, a beta-2 adrogenic receptor, its TM5, or transmembrane alpha helix 5, is extended by 2 helical turns, and transmembrane alpha helix 6 moves outward by 14 angstroms. And this, because of this, because of the extension of transmembrane alpha helix 5 and movement of transmembrane alpha helix 6, a kind of cavity is formed on the cytosolic side, okay, of this G protein copper receptor. So at top of my hand is a G protein copper receptor, and due to the movement of those two alpha helices, a cavity is formed on the cytosolic side of the G protein copper receptor. And now the fifth alpha helix, or one of the alpha helices of the alpha subunit of the G protein is going to dock in that cavity. Okay, so imagine that this is one of those alpha helices of the alpha subunit of that G protein, and it docks in that cavity. That docking, okay, also causes a conformation change in the G protein, okay, So there are two consequences for that docking. When one of the alpha helices of the alpha subunit of the G protein docks in that cavity, two things happen. One is that the bind of GDP is weakened, and the next is that there's going to be a displacement of the alpha helical domain. So the AH domain is displaced. The bonds coordinating the GDP also are weakened, so that the GDP now can escape. And of course, I told you the alpha helical domain is displaced, so that the binding pocket is open. The bind of GDP is weakened, so it can escape. Okay, and this is how G protein copper receptor causes a conformational change in the alpha subunit of a G protein, and acts as a GEF, or guanine nucleotide exchange factor, and promotes the release of GDP by causing a conformational change in the alpha subunit. So that conformational change works like this: that the alpha helical domain opens or is displaced, the bonds or the bind of the to the GDP is weakened, GDP can escape, and GTP, or guanine triphosphate, can get in. There are some conserved sequences in G proteins, both in monomeric and trimeric G proteins, which are very important in the binding of GTP or GDP. And they're called G boxes, okay? And you can see that we have G1, G2, we have 5 of those G boxes. We have 5 of those conserved sequences of amino acids, which are very important in GTP binding. And you can see the residues, the sequence, and the function. And for example, in the H-Ras, which is a monomeric G protein, you can see all of those 5 conserved sequences or G boxes, and also in G alpha I, which is the inhibitory, the alpha subunit of the inhibitory G protein, you can still see those 5 G boxes and completely conserved. Okay, so those sequences completely conserved. And you can see those 5 conserved sequences, or those G boxes, in both an H-Ras, which is a monomeric G protein here, you can see represented in this blue color, and also in a heterotrimeric G protein, the alpha subunit of the inhibitory heterotrimeric G protein. Again, you can see those 5 conserved boxes here. Okay, so I just wanted to show you that we have some G boxes, conserved sequences important, which are very important in the GTP binding. I just wanted to show you that. Okay, so adenylyl cyclase, or adenylyl cyclase, it has a weird name. It's an enzyme. I introduced that, I guess it was in, yes, it was in Biosignaling 1.1. I introduced adenylyl cyclase as the enzyme which is responsible for the production of CAMP. It gets ATP, it removes 2 phosphate, and it turns AMP or ATP into cyclic AMP, which then acts as a second messenger. And so this is the structure of adenylyl cyclase. There are 2 catalytic domains. Okay, so there are 2 catalytic domains, and you can see the adenylyl cyclase here. It's 2 catalytic domains, and this is the the alpha subunit of a G protein activating those 2 catalytic domains. Okay, so this is how G protein or the alpha subunit of a heterotrimeric G protein activates or acts on the catalytic domains of adenylyl cyclase. But I told you that we have 2 types of G proteins. We have a stimulating one, which is the activating one, activates an effector protein, and we have an inhibitory one, which inhibits an effector protein. So now let's see how they work. When we have a stimulatory hormone like epinephrine, it is received by by a G protein copper receptor. And that G protein copper receptor is going to activate a stimulatory G protein complex, okay? And this stimulating G protein complex, when it is activated, its alpha subunit is going to activate or stimulate the adenylyl cyclase to produce more CAMP, okay? So this is I want to call it a mini signal transduction pathway. We have a signal, we have a receptor, and then we have this transducer which couples the receptor to an enzyme, and we have also an enzyme as an effector, and then we have somehow a little bit of a response, which is the production of CAMP. But let's see what happens when we have an inhibitory hormone, such as adenosine. When we have an inhibitory hormone, it is again received by a G protein copper receptor, but now this G protein copper receptor, because it receives an inhibitory hormone, it activates an inhibitory G protein, and the alpha subunit of this inhibitory G protein complex is going to somehow inhibit the adenylyl cyclase from producing more CAMP. Okay, so this is how inhibitory and stimulatory G proteins act on adenylyl cyclase. One of them actually causes activates adenylyl cyclase and causes that to produce more CAMP. The other one inhibits that and from producing more CAMP. Okay, so this is how this is an example of a mini signal transduction pathway in which a G protein plays a very important role and plays a role of a transducer. Now let's examine another pathway in which a G protein acts as a transducer. Again, we have a signal molecule received by this G protein copper receptor. We said that when a signal is received by a G protein copper receptor, it gets activated, and it also activates a G protein. Okay, and now you can see that this is the GQ. Okay, the alpha subunit of GQ protein or G protein Q. This alpha subunit GTP is bound, so this is activated. This alpha subunit, it's going to activate phospholipase C beta. Okay, this enzyme. This enzyme is an effector. So we have a receptor. We have a transducer or a G protein, and this G protein couples this receptor again to another enzyme, like the previous case we studied, couples this receptor to an enzyme, but in this case it is phospholipase C beta. And this one actually cleaves this phosphatidyl inositol 4,5 biphosphate, cleaves this phospholipid into a diacylglycerol and into an inositol 1,4,5 triphosphate, or IP3. Okay, and this IP3, this is the lumen of smooth endoplasmic reticulum, and we know that a smooth endoplasmic reticulum is one of the organelles of a cell which is responsible for the storage of calcium ions. So embedded in the membrane of that lumen of endoplasmic reticulum, we have an IP3 gated calcium ion channels, okay, or calcium release channels. And so IP3 acts as a ligand for this ligand dependent or ligand gated calcium ion channel, and it opens this channel, calcium gets out. So calcium ions then are going to cause the protein kinase C. Kinase proteins, I'm going to talk a lot about them in the next lecture, but kinase enzymes are the enzymes which are responsible for adding a phosphate group to another molecule. It should not necessarily be another protein, but mostly they add phosphates to specific amino acids of other proteins or other molecules. So this is a protein kinase C, and there are once the calcium ion is released in the cell, it causes this release of calcium ions causes the protein kinase C to translocate to the cell membrane. And in the cell membrane or near to it, near the cell membrane, it is the protein kinase C is activated then by the diacylglycerol, okay, also by calcium ions, and one more thing, which is the phosphoinositide or phosphoserine, a negatively charged, I don't remember the name exactly, but a negatively charged phospholipid. It's not shown here, but that also has a role in activating protein kinase C. So in the membrane, it is activated by diacylglycerol and these calcium ions and that phospholipid, one negatively charged phospholipid, and it's going to actually activate other proteins or phosphorate other proteins. Okay, so this is again a mini signal transduction pathway. We have a signal activating, which activates a receptor. It in turn activates a G protein as a transducer. That G protein has an effector, which is in this case phospholipase C beta. It's going to produce some second messengers like IP3 and DAG. Each one of them are going to activate something. IP3 activates a ligand-gated or IP3 gated calcium ion channel, and it's going to release calcium in the cell. And diacylglycerol stays in the membrane and it activates protein kinase C, and protein kinase C in turn activates other enzymes or effectors. So this is somehow the same signal transduction pathway. Shows the production of IP3 and diacylglycerol from phospholipids, and also the release of calcium ions because of these IP3s. I'm not going through all of this, but the reason I'm including this slide as the last slide of this lecture, by doing this, I want to just show you that how complicated signaling and biosignaling and signal transduction pathway can get, and how messy sometimes they can get inside a cell. You can see a lot of different molecules here, a lot of different pathways interacting with each other, and there's sometimes are crosstalks. It's very beautiful, but makes the study of these signal transduction pathways very challenging. But I love it, and so this is just a representation of how messy these signal transduction pathways can get. But one beautiful thing here is that although it is very messy, you can still see you can detect some simple patterns basically. So there is a pattern. So you can identify that, okay, this is happening, these interactions, these chemical reactions are happening in the membrane, so there must be related to the production of IP3, and diacylglycerol. And you can identify this is the lumen of smooth endoplasmic reticulum. You can see those calcium ion channels which are gated by IP3s, and you can see the IP3 here. It's very, very, very small, but it's microscopic, but you can see this is the IP3 acting on these calcium ion channels and releasing these calcium ions in the cytosol and so on. So although they are complicated, but there are some patterns in these very messy and complicated signal transduction pathways. But that was it. That was the Biosignaling 2.1. I talked about the G proteins, two types of them, monomeric and trimeric G proteins. I talked about the regulatory proteins regulating the activation and deactivation of G proteins. In the next lecture, I'm going to talk about a mechanism for switching, which is two mechanisms for switching to common mechanisms. One of them is phosphorylation and the other one is dephosphorylation. I'm also going to talk about different enzymes which are required for the phosphorylation and dephosphorylation. So that was it. I hope you enjoyed the lecture. Thank you so much for watching this video, and I hope you can watch other videos and other lectures as well. Thank you so much.